We're going to move on now to Filippo Castiglione, who's staying up very late to speak to us from Rome. And he's going to be talking about the SARS-CoV-2 infection and cohort study in silico. And I'll turn it over to Filippo.
Thank you, James. Let me try to share the screen. It should be this one. Do you see my PowerPoint?
That looks fine, thank you.
Okay, so let's put it widescreen. Okay, so 15 minutes, I want to tell you a study I have done with the computational model, which is multi-scale, which I'm not going to describe in detail because there's no time. The question I wanted to address with the model was how can we explain the high variability of a clinical presentation of the COVID, basically, because people know that, I mean, from the very beginning, it was clear that the mortality rates was quite diverse in different countries. And there was a very difference in clinical presentations. People get mild disease, others die, and there's no correlation direct apart with the age of the individual. So the question was, can we try to recall this diversity just by focusing on two characteristics of the infection? And one is the host, let's say the health of the host or the immune competence of the host, and the other one is the characteristic of the pathogen itself. So two different competitors and generating such a huge diversity. So basically we use this computational model, I will tell you in a second, to try to test this idea. The model is an agent-based stochastic multi-clonal model with the different cells types, molecular types, interleukins, and so on. And with the many immunological, let's say, hypotheses or processes or whatever functions in it. So it's quite detailed in terms of the biology or the immunology, which is described in the model. So the two components of this study were the level of immunological competence, which we simply represented with a negative correlation with age. So we have this parameters IC, immunological competence, which decreases with age. Okay, it has been a little bit arbitrary, the slope of this curve, but somehow we the aim was to try to reproduce the final distribution of causalties in the age classes, okay, which is not linear. This is important. So this the host factor basically derived two characteristics of the immune response. So that parameter IC was used to determine two other parameters connected to the ability of antigen presenting cell to phagocytize the virus and present. So it's somehow related to the speed of the innate immunity to kick in and start the adaptive immunity. And the other parameter that we somehow tuned with this immunological competence parameter, let me put the laser, was basically the leukocyte count, which in the model is usually represented as a statistical instantiation of this distribution, which are normal range reference in humans, that we basically reduced, like simulating a kind of a lymphopenia due to age. It is well known that there is a relationship between age and the ability of the immune response to, to protect. It's called, in fact, immunological aging. So this concept is coded in this relationship. On the side of the pathogen, we coded two parameters. One is the viral load, the infective viral load. So basically the amount of viral particles at time zero of the simulation. And the other one is the probability for the virus, one viral particle to enter into a target cell. basically the affinity to the ACE2 molecule from the spike molecule of the virus. So these parameters basically identify one simulated virtual individual, and we reproduced 1,500 simulation per age class. So we're talking about a large number of simulations, basically consisting in different trajectories in this stochastic simulation. And then we analyzed the result, and we had to match reality. We had to somehow find a threshold theta, which with which we could classify outcomes of the simulation in three classes. Those belonging to the critical classes are virtual individual which at day 30 of the simulation have still a high level of viral load. So people who have not, virtual people who have not controlled the virus. Then we have the partially recovered class, which is people who have reduced considerably the viral load, but not, cleared completely, while the fully recovered people or individuals are those who have cleared the virus before day 30 of the simulation. So we were observing only the first 30 days, so the initial phase of the acute phase of the infection. Of course, so this is the picture looking at the viremia. We recovered these three different behaviors, and to note the partially recovered class includes the asymptomatic, because those people have a very low viral load, but they are not cleared. So somehow the immune system is coping with the virus, but not completely. And on the side of looking antibody, the same equivalent simulation look like this one. So it's interesting the fact that critical individuals have a very variable level of antibody titers, and which somehow include the class of late responders, people who have, a late, let's say, delayed response to the virus, or people who have a very weak response for different reasons, of course, related to the age and indirectly to the leukocyte counts or immunocompetence, let's say, which is coded in the model. So we we tuned the parameters theta and other parameters I've just mentioned by trying to reproduce the death rate by age at a certain point in history of this pandemic. It was February, I think, or March, something like this. And starting from this, we try to extrapolate and see other statistical outcomes of this virtual cohort. Okay, so we reproduce reasonable parameters. I mean, these numbers here have not been tuned directly. So the fact that in vivo the virus is visible in the pharyngea after 4 days is somehow close to the fact that we have a peak in viral load between 3 and 5 days in the simulation, and so on and so forth. I'm not going into the details, but some of these are very well in agreement to what is observed or at least it was observed in March, April. So the remaining observation are the outcome, and so this is in agreement also with reality, the fact that the viral load correlates with disease progression. This is not a novelty. More virus you get or you are infected day 0, and of course worst will be the final outcome. This is clear for, I guess, any pathogen. The other fact is that IL-6 correlates with disease severity. So the people who have go critical basically show an overall higher level of inflammatory cytokines, and that this is more or less, let's say, constant by age class, so it doesn't discriminate by age class. What discriminates is really different by age class is the level, the type of immune response elicited. Cytotoxic response, for example, it is smaller for aged people, for elder people, than compared to younger people. And interestingly, critical individuals have a stronger cytotoxic response than people who recover. And the reason it seems a cause, but it's a consequence of this fact, the fact that the humoral response is more important in controlling the virus. At least this is what the simulation says. And as you see, those who are experience a critical outcome have a very poor antibody response. And so perhaps the fact that the cytotoxic response is higher is just a consequence. It's the immune system trying to cope with the infection. Many more cells are infected, more possibility for cytotoxic cells to expand. The final result we wanted to see if we could, by looking at the number of cytotoxic cells at day 30 and the antibody titers, if we could somehow predict the outcome of the virtual individual in the class, so if critical or not critical. So we use a logistic regression to split apart these two sets of recovered and critical with its typical machine learning procedure to split these two things. And we found a nice precision and recall score, but only if the antibodies and the CTL, that means the photo taken to the virtual individual, is done after day 25. So before that, the information itself is not sufficient to predict the outcome, the final outcome at day 30. And this is basically what I wanted to say. And I will be happy to take questions if you have. I hope I stayed within the 15 minutes, maybe even shorter. I don't know.
Thank you very much. That was a real taste. People will be hungry for more information, I'm sure. Please type for a question or two.
I have a question if you can hear me.
Sure. Yes.
So I noticed that you model both mortality and the viral load. Do you have, and this is something that I currently am looking for, do you have a profile of when a person dies if they die? Meaning how many days from the beginning? Do you have models for that I can use?
Well, it's not the model, it's an emerging property. The definition of what is dead in my virtual individual is somehow artificial, because of course this is just a model of the immune response. I count cells and cytokines, so I have to set a threshold somewhere and say, and set apart what it's, let's say, in trouble with respect to who is not in trouble. I didn't show it in the slides, but one thing I did was to estimate the damage, let's say, in terms of how many epithelial cells are killed by either by the virus or by the cytotoxic cells. And that is that correlates quite nicely with the mortality, with what I believe is a severe case.
Okay. Do you have several assumptions that can be tested?
Of course, yes. The model can be used in many different ways. Of course, the next step will be to put it side by side with some clinical, not statistical. But the problem is that when you go and look at the specific patient data, it's very difficult to have a very nice fit.
It's all understood. However, if you have a functional form or even a numerical form that says this is what the chances of a person dying in day 1, 2, 3, 4, up until like 20, I can use this. And if it's only an assumption, it's also good. If you have several of those, it's also good.
So
You
could
mean
you,
something like this?
Filippo, maybe you could you share a link to the paper or the preprint describing the model structure?
Yes, please.
Yeah, I will. Well, we still have to submit the paper. I
Ah.
didn't put it in the preprint. I could do it. It will be soon there, probably in a week or something. So
Okay.
Lovely. I think
I will
people,
bother you.
sure.
I think we'd all love to see that.
that
It would be great.
wonderful.
Time for one more question. I hope we can certainly continue conversation offline. Other question? I certainly would like to, I was just love to see the model structure and organization. So that'll be, I look forward to that.
Thank you.
I know it's late for you, so appreciate your taking.
Thank you. Thank you.
Other questions? No?
If you-
Okay. Well,
I have one quick question.
yeah. Gary, go ahead, please.
So this is great work, and I've followed it for multiple years. So fantastic. So have you started trying to simulate hypothetical interventions on this, like interdiction of the various pathways or cytokines that you might have, right, to like test, for instance, monoclonal antibody therapy against inflammatory cytokines and stuff like that?
No, I have done, but it's a very nice. It would be a very nice way to use the model, actually.
All right. Thank you.
Yeah, absolutely. Yeah. Thank you.
Lovely. Thank you again. So
I have to stop the share. Yeah.
So thank you again, Filippo. That was very, very intriguing and again, very relevant to both talks. Thank you to all of our speakers for today. Reminder that this is our last meeting of the year. No meeting on December 24th. I don't think, even with the world which has canceled Christmas, I don't think people really want to have a seminar on the Christmas Eve. Probably New Year's Eve as well. People will be too eager to have the year over to want to have a seminar. We'll begin again January 7th with Paul Backlund and Bart Ermentrout speaking. And then Grace has agreed to speak on the 14th. And we'll have a talk also from Purdue. We're very eager to have people speak. Please contact us if you'd like to speak. You don't have to be a working group member to speak. So if you have suggestions for speakers you'd like to hear, please send those to us and we'll do our best to accommodate those requests. That's been a great seminar series so far. I hope it's been useful for people. Now I'll turn it over to Reinhard